All right, thank you. So I'm going to start with the disclosures for my co-authors for this study, and perhaps include the disclosure that if you hadn't realized, I am not Lynn Abruzzo, who submitted the abstract originally and hoped to be here, but is at home where her 90-year-old mother is recovering from surgery for colon cancer. The research was performed in her lab and designed there. And Carmen Herrling, who was her postdoc at the time, is the person who actually proposed the study, collected the samples, and did a lot of work going through clinical records to make sure they were as up-to-date as possible. The design of what we're doing, we have a training set from MD Anderson, validation cohort from the German CLL8 trial, and the bottom line is that we did this study ultimately with 101 patients from MD Anderson who were assayed using Illumina microarrays. And the validation, we had 109 unmutated cases from the CLL8 trial assayed on Affymetrix arrays. And we looked at the differences between the kinds of samples that we had. And I will point out that roughly the German validation group was somewhat more likely to have higher rise stage, lower beta-2 microglobulin, lower ZAP70, higher CD38, and worse cytogenetics. So building a gene expression signature to look at response to FCR, where we're going across different microarray platforms with slightly different cohorts, was somewhat challenging. I will point out that one of the reasons this is likely to have worked is that we started doing univariate analyses and identified about 1,100 genes that appeared to be associated with time to progression. And we did the standard red-green color map to cluster things, and as we hoped, we divided, using the clustering here, samples into 3 groups, colored blue, orange, and magenta here, and that these had very clearly different time to progression after first-line therapy with FCR. It's interesting to point out 2 facts here. One is that the group colored blue that clearly had the best response does support the idea that many patients with mutated IGHV have very long responses, favorable responses to FCR therapy. But the more important observation for what we wanted to do was the realization that unmutated cases we could clearly divide into 2 groups with very different likelihood of long-term response. And before I go on, I will also point out that we've separated the genes that do this into several categories. And the ones that look most promising here, the largest subsets, we've marked with purple and cyan here, and we'll come back to talk about that in a little bit. The challenge with a sample size as small as this one to build a gene expression signature is that we're not quite sure how robust it is, so there was a lot of statistical work that went into making sure we had a robust signature to begin with before moving to validation. We did extensive bootstrap validation of this to make sure that the genes that we were selecting, the 1,100 genes, were largely reproducible. And moreover, the clustering, well, some strong aspects of it were reproducible. We'll point out here that this plot shows that the blue samples with the very good outcome were almost always classified in that group. The orange samples with the very poor outcome were mostly there, and the magenta ones in the middle, not surprisingly, as always happens with sort of intermediate groups, blurred between the 2. Our conclusion from this is that we were going to build a signature based on what we called reliable samples. That is, ones where we used a standard method that involved all 1,100 genes, which is clearly too much for any realistic clinical test ultimately. We could identify which samples we were highly confident we could always put in the same group at a 90% level or more, and which samples we were less confident in, that they were less reliably classified. We're going to build the signature just using the reliable samples. The plot on the right with the principal components also shows that the ones with the solid colors that were less reliable were not surprisingly on the borderline here. And so we then used the reliable samples to build a model to classify unmutated IGHV patients into an either poor prognosis group or an intermediate prognosis group for their responses to FCR. The genes here, you probably don't recognize most of them because they're answering a different question than what we're used to answering. But the important thing to point out about this is that the resulting signature is mostly a contrast between the purple group of genes and the cyan group of genes. The signs of the coefficients are different in these. The ones in purple, the coefficients are positive, which means greater expression suggests that you're going to have a worse outcome. The ones in cyan are exactly the ones with the negative coefficients, which means that more of that gene suggests a better outcome. When we tested this model again on the training set, including the mutated samples that weren't used in this, almost all of the mutated were in the predicted good outcome group. Again, the boundaries between what we thought were the intermediate magenta and the bad prognosis orange groups were fairly reliable, and even the ones that we couldn't tell, okay, those are the ones that maybe shift between one group to the other. The next step, of course, was to move on to validation, where we had rerunning it on the training sample, looking at cumulative incidence. We clearly get a strong separation between the patients who we predict as high risk and those at low risk. And in the validation group, the separation isn't quite as good, but it's still clearly statistically significant and comes down to at least a year or two's difference in how long we think it's going to take to reach progression. And therefore, we've, again, developed this signature on a small cohort from MD Anderson, validated it on an independent sample from another country, assayed on a different platform, which suggests that there is some aspect to this that is probably real. And in order to understand biologically why this signature might be relevant, we look not just at the 17 genes selected by the statistical method at the end for the final model, but looked at all of the purple gene cluster and the cyan gene cluster to determine what's going on. Certainly the genes in purple were related to higher metabolic activity, especially oxidative phosphorylation, providing the source of energy for the cells. The cyan genes, to some extent, are related to purine binding and some other issues like that. So there's some biological reason to believe the signature as well. And I will finish with a blatant plug, because the paper describing this, if you really want more details, is scheduled to come out in Lancet Oncology in about 2 days. Thank you.
Anybody have any question for Dr. Coombes?
So I have a quick question, Kevin. Have you looked at the intensity of follow-up? Because how frequent is the follow-up is going to change your time to progression between the two groups?
Sorry, I didn't hear.
Did you look at how frequently the patients were monitored in terms of determining their progression? Because
So no, we did not look at that.
and then we have a question now from the public.
Carsten Niemann, Copenhagen. So did you think about how to combine gene expression setup like this with other approaches like recurrent mutations and the known cellular IP factors?
So we did, build multivariate models incorporating many of the traditional clinical and cytogenetic variables. Our model remains significant with that. It mostly replaced some of the cytogenetics and some of the other pieces. Ideally, I'd like a bigger cohort to study that properly.